Just a sort of background to today's meeting, today's colloquium. This colloquium forms actually part of a European project, the EXACT project, and I'll briefly take you through what it's all about. So Amsterdam UNC is involved in network staff exchange, and this is funded by the European Union's Horizon 2020. It's a research and innovation program, and the idea is, as it's in the name already, a staff exchange to make possible exchanges between academic and non-academic partners. So mostly in Europe, but also in some third countries like the USA and Canada. And staff is supposed to be seconded for several months to learn or train on the job, so to say, bring back the information back to the home institutions. And the focus is on integrating precision health in the healthcare systems. So here is an idea of our secondments from Amsterdam UNC. In the past few years, we've sent people to Canada, but also to the Basque region for health technology assessment, and now to the UK. And over the past years, more than 75 secondments have been organized. They take up from 1 to 6 months, and people stay at the home institution that's hosting people from our consortium. So if you have further questions, you can contact me on that email address, as I am the local coordinator of EXACT in Amsterdam. So what is the research focus of EXACT? That's, as you remember, precision health, and to make sure that it's being able to be implemented into healthcare. We have several work packages ranging from health technology assessment to citizen engagement and healthcare professional education. But for today, work package 1 is most relevant. And that's to help define a framework for the integration of big data and digital solutions into healthcare systems. For instance, we focus on state-of-the-art mapping, but also appraising the feasibility of data integration of omics and epidemiological data, and the ethical, legal, and social aspects that are involved.
It's a pleasure to speak with you a bit about what we do at Allelica around polygenic risk scores. So what I'm going to talk about today is some of the work that we've been doing to develop, validate, and critically implement polygenic risk scores from a clinical point of view. And as Dorrit said, please feel free to send in some questions to the chat, and I'm hoping there'll be a bit, there'll be some time at the end to go through those. Okay, so who are we? Why am I talking to you? So as René says, I'm a statistical geneticist. I'm currently Chief Scientific Officer at Allelica. For many years, I was an academic researcher at the University of Oxford, where I'm still based, and where some of our team is based. But here you can see the kind of overview of the team at Allelica. So myself, Giordano, and Paolo started the company now about 6 or 7 years ago, really because we experienced within our academic research this amazing potential for genetic information to be used outside of research, within the clinic, within health. And what we wanted to do was to take some of that research out of the research kind of academic field and into a kind of clinical, into the clinical realm. So we built this company. We've got a great selection of advisors from both the US and Europe, people who are very much at the forefront of implementing genetic information and the use of it within medicine. People like Robert Green, but also key polygenic risk score scientists like Bjarni, who are really developing methods that are really able to squeeze information out of genomes and to allow us to build prediction tools. So Allelica is a software company. We enable labs and healthcare systems to run polygenic risk scores. I'll talk a bit more about that now, but the key thing to kind of note about the company as it stands at the moment is that we're very much a software team. We don't we don't own labs, we don't do lab work ourselves. We very much build software and products that enable labs and actually researchers to be able to analyze genetic data that may be, or that is generated through, through clinical genetics labs. Okay, so I'm sure most of us know what a polygenic risk score is. It's a sum of risk taken from common genetic variants. And here I've just tried to plot the growth of polygenic risk scores and polygenic scores as evidence from a quick search of PubMed over the last 15 or so years. And we can see that there really is a massive increase in polygenic risk score research being pushed out. And a lot of this research now some of this research now it revolves around the validation of polygenic risk scores. So what do I mean by that? So it's one thing to build a polygenic risk score, but in order to enable them to be used clinically, there needs to be some validation of that polygenic risk score. There needs to be some understanding, some validation, some test of whether or not that polygenic risk score has any clinical usefulness. And that's what I'm going to talk about today, is really thinking about how we as a company have built our products based on the really strongest scientific evidence that these polygenic risk scores are actually useful. So the software we build, or the products that we have kind of fall into these three main camps. We have a core set of software called Discover and Validate. This allows researchers, people like Renee, to build new polygenic risk scores or to validate polygenic risk scores on existing data sets. So, current recommendations are that we should try and use as many different methods as possible to build a polygenic risk score, build lots of different polygenic risk scores, and then test those polygenic risk scores on independent populations so that we can have a good idea of what the best polygenic risk score is for a given situation, and then use that downstream. So once somebody has developed a polygenic risk score, we have a separate set of software, which is our Predict software, which is the core bit of our business, really, that allows any lab who can generate genetic data to turn that genetic data into a clinical report, a clinical polygenic risk score report. So we have ways in which those reports are different ways in which we communicate risk through those reports, and I'll talk a bit about that as well. And then we also enable labs to build out, in the US, what's called a laboratory developed test, or an in vitro diagnostic in Europe and the UK, which is a clinical grade polygenic risk score test. So in order to build a clinical test, you need to perform a lot of validation, both from an analytical point of view, but also from a clinical point of view, that allows you to, for a lab to report out a clinical result. And we've performed several of these now, as I say, both in the US and in Europe, and that really is allowing us to present our polygenic risk scores as clinical tests, which is ultimately what we want to be able to do in order to maximize the opportunity that genetics has for enabling prevention and preventative healthcare. So to give you a sense of the kind of people that we work with, I've shown here a kind of subset of some of the different people that we work with. So we work with all of the major sequencing and array providers. We have a partnership with Illumina and Thermo Fisher. We've worked with PacBio in the past to really try and ensure that our software can be relatively agnostic to the input data. As I say, we work with genetics labs, big genetics labs like LabCorp and Invitae in the US, but also European-based genetics centers like MGZ and MGZ is in Germany. Veritas Intercontinental is based in Madrid in Spain. And what our software allows them to do is to offer polygenic risk scores to their customers. So we're really an enabler of PRS testing in that situation. We also work specifically with healthcare systems in the US. So these are people who have physicians who are ordering our tests, providing them to their patients. And we also work with clinical and implementation trials, people who are testing the utility of polygenic risk scores in various different situations, again, both in the US and in Europe and even in Australia. We have a growing interest in working with pharmaceutical companies and biotechs, thinking about ways in which polygenic risk scores can be used outside of kind of pure preventative healthcare, think about ways in which you can use polygenic risk scores to kind of superpower clinical trials, to better power your clinical trial by thinking about the inherent genetic predisposition in everyone to drug response and things like that. And crucially, and I'll probably get onto a bit of this at the end, we're working with payers, i.e. the people who we hope will be paying for these tests at some point in the future, really trying to understand what the evidence is that is required to prove to, in the case of the US insurance companies, that a polygenic risk score is a worthwhile test to fund. And at that point, I'll say actually, one of the great things about working as part of the EXACT consortium is that there are kind of representatives from a lot of these, well, maybe not pharmaceutical companies, but there is representatives, and there is the opportunity to kind of share knowledge across so many of these different types of partner, and in particular think about ways, at least within the European system, that we can build those kind of health technology assessments and other pieces of evidence that are required for implementing, well, actually a load of different preventative tests, but in our case, polygenic risk scores. So I'm going to talk give kind of two examples of where I think polygenic risk scores really are, have some clinical relevance, and I'm going to talk through two scientific papers, one of which was published at the end of last year, one of which is in review now, that we've kind of applied our software and our tools to the problem of trying to build a polygenic risk score, and also tried to document and articulate where and how we think these polygenic risk scores should be used from a clinical point of view. So the two key use cases here are for cardiovascular disease, and in particular a subset of cardiovascular disease called coronary artery disease, which is basically heart attacks, and then breast cancer. Okay, so when we think about developing multi-ancestry polygenic risk scores, we need to do the following things. We need to compile diverse data sets. So this means compiling data sets with genome-wide associations, summary statistics or validation or testing cohorts that contain individuals from a broad range of different ancestries. So why is ancestry important? Well, one of the key criticisms that is a completely valid criticism about polygenic risk scores and actually genetic analyses in general is that they've really been targeted on or based on data from European individuals. That's because most of the genetic data out there is from European ancestry individuals. So what we want to do when we think about implementation and translating research into the clinic is ensuring that we have polygenic risk scores that can be applied to any individual regardless of their genetic ancestry. So that's why we've kind of concentrated on this multi-ancestry component. So we compile these diverse data sets, we then can develop and validate either ancestry-specific polygenic risk scores or at least test and evaluate a polygenic risk score on different ancestry groups, so that we can really ensure, as I say, that we have polygenic risk scores that can predict risk in a range of different individuals. And then once we have those polygenic risk scores, we want to really think about how we can demonstrate clinical utility, then demonstrate how these can be implemented within a clinical setting. So there are two key assumptions that I want to kind of have in everyone's head when we talk about these things. And the first is that if we're thinking about ensuring that we have a polygenic risk or a genetic test more generally that is applicable to any patient or individual that comes into a clinic, we want to make sure that there are PRSs that can be used on anyone. And the way that we think about that at Allelica is to ensure that we have optimized the available polygenic risk scores to be applicable to anyone. And the way that we think about that is to optimize polygenic risk scores to individuals with similar genetic ancestry or genetic similarity. And another key component, I think, just to kind of face off a bit of challenge that often is presented to people who build polygenic risk scores, is that we're really not in the business of suggesting that polygenic risk scores should replace any kind of clinical risk assessment test. And in fact, the clinical utility of polygenic risk scores really can be best achieved when polygenic risk scores are combined with other known risk factors, and also allowing for the fact that there is some correlation potentially between these risk factors, but actually that polygenic risk scores can be shown to identify reasonably orthogonal levels of risk to these other clinical risk factors. So when we think about implementation, what we're thinking about is trying to ensure that as many of the different tools that are available to predict risk are there, of which PRS is one, and that is really where we're going to achieve the best clinical utility. So we know that genetic variation influences disease risk for a range of different diseases. Here's an example from breast cancer, where at the left-hand side of this plot, you have the kind of low frequency, rare pathogenic variants that have a much bigger impact on disease and the much more common variants down on the right-hand end here. So there's this kind of well-established relationship between the frequency of a genetic variant and its effect on disease. And what genome-wide association studies have really allowed us to do over the last 15 years is to really identify many, many more of these common low penetrance risk alleles here down in the left-hand corner, but also to get decent estimates of the effect of those common risk alleles on disease. And that's really what we're leveraging when we build these polygenic risk scores. So when we build a polygenic risk score, the general process is as follows. We want a discovery data set, as I say, a genome-wide association study, where we have for a range, typically millions of genetic variants spread across the genome. We have some assessment of their effect on disease, of their beta, the effect size of disease for an individual that carries a risk allele. So that's really the bread and butter that goes into these PRS development step. Key thing is that there are now increasingly more and more methods that can be used that you can leverage multiple different genome-wide association studies from different cohorts, different ancestry cohorts. So we really have a lot of raw data at our fingertips to build polygenic risk scores. So once we have these genome-wide association studies, we want to try, as I said at the top, many different algorithms to really try and identify a best performing score. So there are different algorithms that leverage different kind of aspects of genetic data. Some of these algorithms adjust for linkage disequilibrium, others threshold the p-value, the GWASs by a p-value, and some also, then there's some Bayesian methods that think about incorporating linkage disequilibrium to shrink down the effect sizes of particular variants. Regardless of what the actual kind of mechanics of these different algorithms is, ultimately what this step of the PRS building process is aiming to do is to apply as many different algorithms as possible to identify many different panels, and then from that set of panels, identify the panel that has the best performance as measured in a variety of different ways, but the best performance on a validation data set. We then want to take that best performing score and apply it to a brand new testing data set containing individuals who have not been used at any stage in the past to ensure that that polygenic risk score has decent predictive performance on an independent data set. We want to make sure that the polygenic risk score is not overfit to any of the previous populations. So once we have that, we've built this polygenic risk score, we have an assessment of its predictive performance on a testing data set or even a range of different testing data sets. So these testing data sets could comprise individuals of different genetic ancestries, for example. We can have we end up with a polygenic risk score panels, a set of variants, risk alleles, and effect sizes that we can then use to predict risk in our individuals or patients. So some key properties of polygenic risk score panels are that they typically comprise hundreds to millions of common genetic variants, depending on which one you use and how it's built. As I say, there are various different algorithms that can be used to account for different correlations and effects by nearby variants. So what we're really trying to do is to pull out from the genome-wide association studies, the really kind of the true effect sizes for these variants, remove a lot of the noise. And then, it's key to note that actually at an individual level, typically polygenic, the individual variants within a polygenic risk score don't have a big effect, but you get the effect of a polygenic risk score by summing these variants together. So now we've got a polygenic risk score panel. We can apply it to a population of individuals, and what we tend to find is that these polygenic risk score the polygenic risk score, which is just the sum of the effect allele, the effect from the number of effect or risk alleles in individuals carrying. So you get an individual value for each person in the data set. When we apply this to a population, We get this normal distribution where most people have a score somewhere in the middle, some people have a very high score, and some people have a very low score, but you get this nice normal distribution of these values. And what we want to see is how that polygenic risk score relates to risk. So in this population, if we know what the outcome is, if we know who has got disease and who hasn't got disease, we can then split that population into bits, into parts. Typically, we think about percentiles, so hundredths of the distribution. And then we can look in our data set of known disease at what the risk associated with being in each of those percentiles is, or what the incidence of disease is in each of those percentiles. And if that polygenic risk score is doing a good job of predicting disease, we find that individuals who've got higher polygenic risk scores, so they're in the higher percentiles, will have greater lifetime risk than people at the lower percentiles. So that's what we really want to be able to do. And what we want to show from a polygenic risk score is it can, in as many populations as possible, accurately predict risk. We can then go back to our original risk distribution, identify tails of the distribution where individuals might have a certain increased risk of disease. So in this plot here, it might be that end of the distribution, that tail of the blue tail of the distribution has twice the risk of the rest of the distribution, or the gray part might have 3 times increased risk. But what we're able to do here is to kind of go past just an assessment of what an individual's PRS value is, or what their percentile is, but really translate that into an estimate of what that polygenic risk or value has, the effect of that value has on disease risk. So this is a figure from the Nature Communications paper that I kind of mentioned right at the top. I just wanted to kind of walk through what we do here what we've done here in terms of thinking both about ancestry specific kind of polygenic risk scores and ensuring that we have polygenic risk scores that work equally well on everyone, but also thinking about how we can use them clinically. So in this top left quadrant of this plot, we can see there are 5 different groups of odds ratios here. Each one is a set of odds ratios of the polygenic risk score applied to different ancestry groups. So this is an African group at the top, then an admixed American group, an East Asian group, a European group, and a South Asian group. And across all these groups, we've applied these polygenic risk scores to these held out testing data sets, which are either the UK Biobank or the MESA cohort, which is an American-based atherosclerosis cohort. And we can see that across these different cohorts, either on their own or combined as a meta-analysis, we get good prediction from the polygenic risk score of disease risk. So we're getting an odds ratio of at least 1.5 across these different groups. When we try and think about how that translates to risk in the way that I kind of described just now, think about the tail of the distribution where you get individuals at a certain higher risk, we can use those polygenic risk scores to identify a tail of individuals where the tail here has 2 times increased genetic risk to the rest of the population. So we selected that risk threshold, that 2 times risk threshold, because actually it's equivalent to what can be considered a risk factor for cardiovascular disease. So things like having diabetes, having a high LDL cholesterol can increase your risk by 2 to 3-fold compared to the rest of the population. So identifying people who have genetic risk, having controlled for all these other factors, but who have genetic risk that puts them at a 2 times increased risk against the rest of the population is really a kind of clinically actionable level of risk to be able to communicate people. And in this bottom set of figures here, I'm showing how we can use that information of using the polygenic risk score as a risk factor to reclassify individuals, and how that reclassification of risk based on the presence or absence of a genetic risk factor leads to a better prediction of who goes on to get disease. So in this bottom plot here, we've applied something called the pooled cohort equations, which is a clinical 10-year risk cardiovascular disease risk prediction model, which puts individuals into one of 4 categories: a high category, which is over 20% 10-year risk, an intermediate category, which is somewhere between 7.5 and 20% 10-year risk, borderline, which is 5 to 7.5 10-year risk, and low, which is a less than 5% risk. And so in each of these groups, you can see that, as expected, individuals who have low clinical risk factors, as estimated from this pooled cohort equation, will have low number of observed events in this prospective cohort. But as we assess clinical risk, the overall risk or the overall observance of events in these groups increases, which is what we'd expect. Individuals with higher clinical risk will have more events. But in each of these, we've stratified individuals based on whether or not they have a genetic risk factor or not. So individuals who have a genetic risk factor are represented by a square. Individuals who don't have the risk factor are represented by a diamond. And if we don't take into account the risk factor, you're the circle. So what you can see is that when we take into account the risk factor, particularly in this intermediate group, we can see that those individuals who have a genetic risk factor here have a much higher incidence of disease than people who don't have the risk factor. So what is this telling us? It's telling us that there is additional information on top of that can be gathered from a clinical risk assessment that can be used to identify people who are at higher risk. And actually, if we think about how this can be implemented from a clinical point of view, there are guidelines both within the US but also more generally in Europe that allow for the kind of the recognition of risk factors, additional risk factors that can be taken into account over and above an individual's clinical risk. And one way in which polygenic risk scores for coronary artery disease can be used to augment clinical risk is to say, look, have you got intermediate clinical risk and then a genetic risk factor as identified through a polygenic risk score? If you have that genetic risk factor, your intermediate risk should potentially be up-classified into a higher risk group. Once that is done, there are key recommendations about what to do when you're at high risk. So individuals who are automatically at clinically high risk, at least in the US, are pushed onto lipid-lowering drugs like statins. So one argument for using, one way in which polygenic risk scores could be used in this scenario is as a risk-enhancing factor and a way of reclassifying an individual's genetic risk, clinical risk on the basis of their genetics. So now just to move across to breast cancer, and again, this is based on a publication that we've got under review at the moment. Here I'm just showing across different ancestry groups here. So we've got Asian groups, African and European groups, some assessment of the predicted performance of these different ancestry-specific polygenic risk scores. And across these groups, we see good predictive performance from the polygenic risk score. Odds ratios of greater than 1.7 in each of these groups. So that means that we can use polygenic risk scores to really identify people at increased risk of breast cancer. When we use those polygenic risk scores that we can show have a good predictive performance and apply them within a clinical risk assessment model, in this case, this is the Tyrer-Cuzick model. It's a clinical risk assessment model that can be used to identify people who are at high risk of breast cancer. We can apply the Tyrer-Cuzick model, which just takes into account clinical risk factors, or an integrated polygenic risk score and Tyrer-Cuzick model, which takes into account clinical risk factors and the polygenic risk score. We can compare the ability of those two different models to identify people who are at high risk in a prospective cohort where we can follow people and understand who goes on to have disease. We can understand which is a better classification of risk, which one is more accurate. And this plot shows here across these different ancestry groups, with the exception of Asia, that implementing a polygenic risk score within this Tyrer-Cuzick model leads to better classification. So we've got positive values here, positive net reclassification improvement, which is an assessment of how much better a model does than another model. So the model with PRS has got a better classification of risk across than the one that doesn't take into PRS across these different ancestry groups. And the different kind of columns here just show different time horizons and different risk strata or different ways of estimating risk. And we can see that, for example, on the leftmost column here, we use the Tyrer-Cuzick model to assess 10-year risk and then classify people as to whether or not they're above a 2.5% 10-year risk threshold. That's a risk threshold that is used to identify women at higher than average risk. We do a much better job of, potentially up to a 10% better job of accurately identifying risk in those individuals when we incorporate the Tyrer-Cuzick. So as I say, each of these different time horizons and risk horizons shows a pretty similar story. that incorporating the polygenic risk score within this clinical risk model leads to better and more accurate risk assessments. So how can we think about using these results within genetics labs and health systems? So what I've kind of been speaking about up till now is largely kind of the research that we have been doing, and is actually, supported by research that others are doing that really show that there is some clinical utility to understanding a polygenic risk score. But practically, how can we think about implementing these polygenic risk scores within kind of clinics? And actually, whilst kind of conceptually generating a polygenic risk score is a relatively straightforward thing, you want to just get some genotype data, you want to get the PRS panel, you want to work out from the genotype data how many risk alleles somebody's carrying, and then apply, the effect size of each of those risk alleles to that individual, and then sum together all of all of that risk, all of that effect associated with their risk alleles into a single value. Actually trying to think about how to build robust and scalable pipelines that can implement this from a clinical point of view, which is all documented and as I say, reproducible, is actually a big challenge. It's a big challenge for multiple reasons. One is that you of course want to make sure that the actual genetic assay, the polygenic risk score that you're using, is fit for purpose. It has some clinical validation. But there are also a bunch of different steps, both bioinformatic and also kind of practical, that need to be taken when thinking about just taking that polygenic risk score and implementing it in a robust pipeline. So here I'm kind of showing some results of a project that we're working on with Baylor College of Medicine, who are implementing our polygenic risk scores within a clinical genetics lab based on whole genome, high coverage whole genome sequencing. There are a range of different things that need to be taken into account. Things like ancestry adjustment of the polygenic risk scores. So within an ancestry group, even applying a polygenic risk score within that group needs one to require about needs one to think about fine scale ancestry issues, think about how allele frequencies at these common variants will vary based on underlying ancestry and not just disease risk. So there are ways to correct out potentially spurious estimates of risk that are based on population history or ancestry and not on disease risk. We'll go into a bit of that in more detail at the end if there's time. But there's also ensuring that the risk that is applied, the risk model that is applied to an individual on the basis of their polygenic risk score value, has some clear paper trail, has some clear estimate of risk, so that when that PRS value is converted into an estimate of risk, we really have an accurate estimate of what that risk is. And then, of course, there's like a bunch of different things and analyses that need to be done to show that the genetic data that's being generated by a lab is being done in an accurate way that is reproducible. So this requires building up a framework for running truth samples through a lab to ensure that individuals who what the PRS value is, you have DNA from them and you run them through a lab and compare to the truth, having run everything through the pipeline. And then, ensuring that all of that validation, both from a clinical point of view, but also from an analytical point of view, is robustly done and recorded so that you can build out a lab infrastructure to run these polygenic risk score tests. And then, of course, there's the last step here, that is applying that polygenic risk score to a new individual. How do you ensure that, as I say, that PRS value that you estimate is as accurate as possible? But then thinking crucially about how you report out genetic risk and what the information is that you're providing somebody on the basis of this genetic test. So the really nice thing is that we're not the only people thinking about this, and in fact there are a couple of reasonably recent papers where people have other people have articulated pretty much the similar sort of ideas that we've been working on at Allelica now for 6 or 7 years. There was this paper from a couple of years ago in Nature Medicine that really kind of unpacked both all of the kind of statistical and epidemiological analyses that I've been talking about, but thinking about how do you make sure that a lab is able to run these tests? How do you ensure that there's validation and robust processes in place so that polygenic risk scores can be implemented as a clinical test? But then, of course, thinking about how to report out clinical risk and think about ways in which that information can be used to to lead to action, which is ultimately what we want to do here. Another recent paper, this is now from just about a month ago, again in Nature Medicine, from the Broad Institute, based on the Emerge project, really kind of did some further kind of articulation of many of the things that, as I say, we've been working at Allelica now for several years. Things like thinking about the actionability of the information that you get from a polygenic risk score, ensuring that ancestry is properly accounted for, and ensuring that the risk is reported in a way that means that there is some actionability to that. So if anyone's interested in really unpacking the kind of all of the kind of deep science that goes into the validation, then both this paper and the previous one are really worth a read. So just to kind of go over again in a bit more detail how we do this, just to kind of really hammer home the fact that it's a reasonably complicated pipeline that needs to happen, but one that is totally solvable by software. I'm just going to kind of run through a kind of classic pipeline of how we get from genetic data, in this case microarray data generated from an Illumina iScan machine, into a polygenic risk score. So you get from that machine, you get an IDAT file. That's just a measurement of intensities, and then within our software, we then perform a variety of different quality control, a variety of different tests on that individual level IDAT data. It's turned into a VCF. Imputation is typically done then from that data set, that kind of clean imputed data set. We can estimate principal components. We can estimate what an individual's genetic ancestry is, and we can use those. We can use those to do several things. The first is to ensure that we do a sensible adjustment of that polygenic risk score data based on an individual's ancestry. Sorry, this is my transition. Gone a bit funny here. But we can also use that genetic ancestry to identify the appropriate risk model to apply to an individual. So this is really important here because we know that polygenic risk scores will perform differently on different populations, and that shouldn't be a barrier to using them, but it should be something that is really kind of taken into account when you're translating that polygenic risk score into risk. You want to know, you want to find, from your set of testing data, your set of PRS models, you want to find the distribution that is most similar to the individual whose risk you are predicting, and so that when you get that value and you turn it into an assessment of risk, you've really got an accurate assessment of what that risk is that is coming from a population of individuals that is similar to that, the patient that you're testing on. So as I say, once you've done all of this sort of nitty-gritty work, really trying to kind of ensure that the PRS value that you've got from an individual is as accurate as possible, accounted for as many different things as possible, and you've applied that to a risk distribution, a risk model, you then convert that into the estimate of risk, and you want to report out that risk. So how do we convert that PRS into risk? Well, one way is to think about relative risk. So as I said at the top, we have this distribution of risk. This is a distribution of a breast cancer polygenic risk score. And here, if somebody's in the 90th percentile, that means their PRS value would be about there in this plot. So how do we turn that into an estimate of risk? Well, we go back to this idea of splitting the distribution into percentiles. You split that distribution into percentiles, 100 different equally sized bins. And in each of those bins, you can estimate what the relative risk is here relative to the middle of the distribution, the median percentile. You can work out what an individual's relative risk is based on their genetics there. And then you can say, right, well, at the 90th percentile, this individual has an over twofold increased risk compared to the median. So that's how we can think about changing, translating the polygenic risk score from that distribution into an estimate of risk. The crucial thing here is that that relative risk estimate is based on applying a polygenic risk score to a known testing population that has people, in this case, with and without breast cancer. So you really have a good sense of both what this polygenic risk score can do in terms of prediction, but also ensuring that that population is of a similar genetic ancestry to the individual that you're predicting risk from. Okay, so I'll try to be quick over the next 5 or so minutes, just to kind of wrap up how we think about this for breast cancer and cardiovascular disease from a real kind of clinical point of view. So breast cancer, it's the most commonly diagnosed cancer in women across the globe. Here are some numbers. There's over 2 million new cases every year. These are numbers from 2020, almost 700,000 deaths. It's a real important burden, highly, high burden on society. And actually what we know from these these analyses of incidence figures and things is that over the past 20 or so years, whilst incidence levels have gone up, mortality levels have gone down. And the reason is that we're capturing disease earlier, treating it earlier, and that's really driving down mortality. But we could still do a lot better. And one way in which we could do better is to have better risk assessments. So there are already clinical risk assessments that identify people at high risk. So I mentioned the Tyrer-Cuzick model. That's a popular one used in the US. There's also the Gale model, Boadicea model, or Canris model that can be used to identify a woman's 5-year, 10-year, or even lifetime risk of disease. We also know that there is, for breast cancer, monogenic testing that can be used to identify people who are carrying rare pathogenic variants. And we know that they are associated with certain increased levels of risk. And the crucial thing about breast cancer, and the nice thing about breast cancer, is that there are already widely established national guidelines that say if your risk is at a certain level, you should have more mammograms or more sophisticated surveillance like MRI, or you should start screening at an earlier age, etc. So there are clear guidelines about what to do when you're at high risk. So the question we want to answer is, how can we use polygenic risk scores to get better at those risk assessments? And this is just a kind of example of some of the guidelines, one set of guidelines from the US. This is the NCCN guidelines. And here you can see that if an individual's at average risk and they're over the age of 25, under the age of 40, then really they can just go to their doctor, have a kind of chat about it, but there's not really a lot that can do in that average risk group. However, if you're at increased risk, if you're down here, there are various different ways in which mechanisms can be put in place to essentially mitigate that risk through increased surveillance. So how do we use Allelica's products or our reports to communicate breast cancer risk in this kind of paradigm of thinking about high risk? Well, there are a number of different ways. As I say, we can integrate them into risk tools. We can also use them as a way to identify what an individual's overall genetic risk is, at least from common genetic variants, and how that translates into a lifetime risk of disease. So here in the middle, you can see a woman who has a high polygenic risk score. She's right in the tail of the distribution. She's in the 98th percentile here. And that equates to an over 25%. It's a 30% lifetime risk here. So she's right up in the top of the distribution. And because that's over a guideline risk threshold of 20 or 25% high lifetime risk, that information can be used to allow her to potentially access increased surveillance, as I say, and to better manage that higher risk. We can also think about when using this sort of information to identify women on whom earlier screening might be more beneficial. So national guidelines dictate that there is an age at which women should start screening. That's typically the same age for the whole population, unless you're known to carry a rare pathogenic variant, but that's going to be a very small proportion of the population. So one way in which we can think about using polygenic risk scores, but also risk models in general, is to say, when does a woman approach the population risk at the screening age? And how much earlier in life does she reach that on the basis of this increased risk, and use that as a way to indicate when a woman should start screening? So here you can see that this blue line represents a kind of average population risk, 10-year risk, and you can see that this is at age 50. The screening threshold of this equates to a lifetime, a 10-year risk of just over 2.5%. But this individual, this woman, because of her high polygenic risk score, has a much higher, much greater overall 10-year risk trajectory over her age. But also she's reaching this 50-year threshold much earlier on in life, potentially 10 to 15 years earlier. So the repercussions of this genetic test is that this woman should potentially start screening earlier than the national guideline threshold of 50 years. It's also the case, and again, I push people towards looking at the preprint that we put out, that we can use polygenic risk scores to both identify women who are at equivalent risk for carriers of rare pathogenic variants in breast cancer. So here we can see this group of dots down here representing different ancestries based on our polygenic risk scores, and the increased risk associated with being in the top 5% of the population here. It's all hovering around a threefold increased risk for that top 5% of the population. And actually that's equivalent to the average risk for carriers of many of these rare pathogenic variants. But as you can see by the size of these points compared to the size of these points, that the frequency of people in the population with that increased risk from their PRS is much, much greater than the risk of carriers of these pathogenic variants. So we're really able to show that PRS can identify women who are at equivalent risk to carriers of many, not BRCA1 or 2, but some of these less penetrant pathogenic variants, and really opening up the potential for polygenic risk scores to really identify more women who are at equivalent risk to some of these monogenic variants. I'm going to skip over this in the interest of time, because I know we've only got 10 minutes left, but just to say that we've kind of also been thinking quite deeply about how to implement polygenic risk scores for cardiovascular disease. And the key takeaway here is that actually when we use genetics to polygenic risk score to identify people at higher risk of cardiovascular disease, it really does, from both a cost effectiveness point of view, so this is a cost effectiveness analysis, indicate that, by doing that thing, as I showed in that figure from the paper, by doing that thing of reclassifying individuals at intermediate risk into higher risk, and therefore pushing them down a kind of high risk clinical pathway, it both reduces the burden of disease on patients, but it's also cost effective because you're having fewer people going through and having adverse effects, expensive effects, heart attacks and things. You're reducing down the overall cost of the disease on health systems by decreasing the number of people who go on to have events by targeting interventions on those who are at higher risk. And as with breast cancer, and actually all our polygenic risk scores, we've also built these different ways of communicating risk to ensure that there is some actionability, both in terms of the level of risk that we identify people at, but also trying to gauge and ensure that the way that we present this information is done in a reasonably transparent and straightforward way. Okay, so I've spoken about how development is moving very fast. It's moving, incredibly fast and faster every year, and many of the perceived barriers to implementation are rapidly being removed. PRS is getting more and more powerful at identifying people at increased risk of disease. They can be more accurately applied to individuals of diverse populations. So we're removing the perceived barrier of the non-ability of polygenic risk scores to be applied to different populations. And it's also, I hope, clear from what I've been saying that actually one way in which polygenic risk scores and genetic information in general can be used in the very short term is to integrate them within current risk prediction paradigms and pathways. So I've showed the use of clinical PRSs for the management of breast cancer, how we can improve clinical risk assessments, how we can stratify women, stratify risk in women who are carriers of pathogenic variants, but also identify women who have equivalent risk from their PRS to carriers of those pathogenic variants, and also think about, although I didn't go into too much detail about this, about how we can use a CAD polygenic risk score as a risk enhancer for cardiovascular disease. Instead of that, there are clearly challenges that still remain. For example, using these at a population level, thinking about reimbursement and who's going to pay for these polygenic risk scores is a really key and critical kind of step forward to the implementation of these tools. Education, this is a big part of what I spend my life doing, is talking to people, trying to explain to people what the benefits are, what the limitations are to polygenic risk scores. But this needs to happen both to physicians and patients in the public. So we have a kind of a side project within Allelica called our Clinical Academy, where we get experts to present webinars that are hosted on our website that provide kind of information about the clinical use of polygenic risk scores. And there's also kind of key questions and key work that needs to be done to understand how best to regulate polygenic risk scores. You know, we leverage very strongly the laboratory developed test, the LDT pathway in the US and the IVD pathways in Europe. But you know, within those pathways, there's definitely still increased opportunity and increased need to ensure that the way in which these tests are presented clinically, you know, aligns with what the regulators want. And I think a lot of what we've been doing at Allelica, within all of the clinical validation and all of the analytical validation, is really trying to ensure that, you know, these things are used and in a way which it should be used, and you know, limitations are upfront. But you know, ultimately, what we're trying to do here is to produce information that can be used, you know, powerful information that can be used when, you know, to really improve the clinical outcomes of patients. Sorry to rush quite a bit at the end there, but if there are any questions, I'll take them now.
Yeah, thank you very much, George, for this wonderful presentation. Very insightful. I saw in the chat that's a question by Jeff Beck. So maybe, Jeff, you can pose it directly, or...
Sure, I can do that. I didn't know if there was going to be a backlog of questions, so figured I'd enter one in. But yeah, I'd like to reiterate Renee's sentiment. George, thanks for the wonderful talk. I had two questions for you. Firstly, regarding the reporting of PRS results, does Allelica provide the information directly to patients, or is it reported or mediated through a healthcare provider like a physician or genetic counselor? And then secondly, what are your thoughts, arguments for or against including this information into a patient's electronic health record?
Yeah, great question. So the answer to the first question is that yes, all of our polygenic risk scores are initiated through a physician and are communicated through a physician or through some genetic counselors within a physician's office. So in order for our tests to kind of be treated as a clinical test, there needs to be a kind of clinical oversight. And therefore, the way that we do that is to ensure that a clinician orders the test. At the moment, the test is still paid for by the patient. So there's a kind of a step there that requires an individual to, you know, a patient to pay for the test themselves, but there's always clinical oversight, either, as I say, directly through a clinician or through genetic counselors working on behalf of that clinician. Yes, so should this information be put in the electronic health record? We do work on integrating information into the clinical health record. I think that what I would greatly, I would really advocate for putting this information into an electronic health record. The challenge with that is not necessarily from a technical point of view; it's more from a kind of educational perspective, and you know, ensuring that if this information is put into electronic health records, then any physician who is accessing that has some idea of what that information uses, and it doesn't just lead to confusion as opposed to you know, helping or aid. So I think that they're in my mind anyway, but if anyone else has any other arguments, it'd be great to hear them. But I think it's more an educational problem there, and also ensuring that any polygenic risk score that is put into a health record has some, you know, clinical or health-based reasoning for putting it there. You want to have information that's relevant to an individual's health, not just, you know, some of these polygenic risk scores that can be used to kind of predict hair color and things like that.
Speaking to that point, then how do you advise, you know, because PRSs are continually being refined with powers, or the, you know, improving power of GWAS and better validation, as you alluded to. How do you advise then adjusting that value in the EHR if a new GWAS comes out that improves the prediction capability?
Yeah, so the way we're thinking about this at the moment is to sort of have a versioning and an annual kind of update. Again, and this is actually a very kind of valid criticism or challenge about polygenic risk scores, is the fact that they can change over time. And like you say, they're typically changed because the models get better and more powerful. But what that can introduce is uncertainty that the test is doing what it says it is. So, it can lead to an individual being reclassified, as the test is improved. So they might be at high risk on one version of the test. You then put out a new version, and the risk gets re-estimated, and now they're not at high risk anymore. And so, at an individual, and this is a big challenge, is how do you're showing, in order to show that, in order to improve the test, you're showing better performance at a population level. But at an individual level, that means that there can be some kind of movement around within the risk. And I think an important part of dealing with that is to kind of embrace the fact that these are dynamic, and to try to not scare people that they are going, that your risk might change over time. But that is kind of an inherent part of this process. But I understand that there are kind of valid criticisms about that being perceived as something, as potentially being your test being inaccurate at the individual level because of this reclassification. But as I say, I think the way that we're thinking about it is to kind of update tests on a reasonably regular basis, and then ensure that there is communication around the fact that if a result or classification changes over time, then that's not a bad thing. It's just because these tests are getting more accurate.
Okay, George, you maybe have time for one more question?
Yeah, sure.
Because I saw Dorette raising a hand.
Yeah, well, first of all, thank you very much for a wonderful comprehensive talk. I had maybe a quick question. I noticed that you switched sort of between lifetime risk and 10-year risk.
Yeah.
Does that depend on the context, or whether you're talking more general or to an individual patient?
Yeah, so that's typically to do with how we want to. It's typically to do with with guidelines. So, are there any guidelines that say if your 10-year risk is above a certain value, then you should do X, or if your lifetime risk is above a certain value, you should do Y? And so what we've tried to do is to ensure that the risk that we communicate, whether that's a relative risk or a 5-10 lifetime risk, aligns with some sort of guideline recommendation on what to do with 5-10 lifetime risk. So that's really what is driving whether which level of risk we report. So, for example, in the US, 10-year risk is used to 10-year risk of breast cancer is used to identify whether or not a woman should be put on tamoxifen. Whereas lifetime risk is used to understand whether or not a woman should start screening at an earlier age. So we kind of try to, think about ways in which the information that is output from a PRS or a PRS integrated model can align with some sort of recommendation based on that risk.
Thanks.
I see one more question. Do you have time, George, or should we?
Yeah, the one from Carla.
Yeah, the one from Carla, yeah.
For CAD, in the intermediate category, it seems to shift towards a higher risk when adding PRS. Could this be a priority to applying PRS? And did you assess downgrading risk and its consequences? Yeah, so absolutely. One of the ways that we are really thinking about kind of, rather than applying a polygenic risk score to everyone, is to say, right, there is a group of people who are at intermediate risk. At the moment, the guidelines in the US at least say, if you're at intermediate risk, you should take into account other risk factors, risk enhancing factors, things like actually ethnicity or diabetes or high triglyceride levels and things like that. And then you have a consultation with your physician or about what the best way to kind of mitigate that risk is. So we would absolutely advocate that PRS should be one of the additional pieces of information that can be used there to as a risk enhancing factor that says, right, if you've got intermediate risk, but you've got several of these risk factors, of which PRS might be one, then actually you should start maybe going down a more aggressive risk reduction route that might involve drastic lifestyle and dietary change, but could even kind of instigate lipid lowering drugs or more aggressive lipid lowering drugs. So I think that's the nice thing about that is because particularly when you look at the way in which payers want this information provided, and also health systems, is trying to like work out what the right set of individuals is to apply a test to. And just saying we should apply it to everyone is sort of not really good enough. You kind of want to kind of identify a group of people, and actually I'd argue that group of people who've got like some clinical risk, and that could be pushed up by a genetic risk, are a really good population on which this test could be used.